Alright, sorry I haven't done the screencast earlier, but my computer's in the shop, so here we go. Replication is the process where we take DNA and we make a full copy of all of our DNA, all of our chromosomes, when we want to make a new cell, either through meiosis or mitosis. So we need to copy all of our DNA first. This process occurs in the nucleus. Transcription, another process that occurs in the nucleus of eukaryotic cells, would just occur in the nucleoid or in the cytoplasm of a prokaryotic cell, is where we take DNA and just one gene, we transcribe that gene into a copy of messenger RNA. And so this process is different in the sense that we only need one gene, one segment of DNA, and we transcribe it into messenger RNA. Translation is we take that messenger RNA, the copy of the gene, and we translate it into a polypeptide, a chain of amino acids, which we would fold into a protein. And so that messenger RNA is translated into a protein, and that occurs in the cytoplasm, outside of the nucleus. And a ribosome is what allows that to happen, whereas transcription, the enzyme that's involved in transcription that makes messenger RNA, is RNA polymerase. The enzyme involved in replication is DNA polymerase, and so ribosomes are involved in translation. So I took the lines and I broke apart the DNA into the little 3 nucleotide sequences that we refer to as triplets. Now the triplets obviously correspond to codons, and which then correspond to anticodons, but it all starts with the DNA, and those 3 nucleotides, or that 3 nitrogen base sequence, is called the triplet. Now, number 3 you got to read carefully. Remember, it's asking still for DNA and the complementary strand across from that DNA. So it's not asking you to do any translation, or transcription. It's asking you to, what in the world is going on here? There we go. I don't know why I highlighted everything there. All right. Probably because I have the highlighter selected. So across from the DNA is going to be, like you said, across from C's or G's, G's or C's, A's or T's, so you know it's DNA because you still have the T there. And there's another T, that's how you know it's DNA. And I can just delete that like that. And then, there we go. And across from A's or C's or G's, A's or T's, and T's or A's, so on and so forth. Number 4, we have transcription. This is using this sequence up here, right? Sequence from, well, I'll just highlight that right there. Okay, that sequence, plus these 3 over here. So we're going to use that sequence in order to do this transcription right here. So these are going to correlate right here. So if you look at these are all going to correlate right here. That is going to correlate with that, including this. I don't know why that's not highlighting here. Problematic, as always, as all technology is. So anyway, across from C's or G's, and G's or C's, but look, across from A is not a T, it's a U. So it's pretty much exactly like the complementary base strand, except in the place of T's or U's. So there is the sequence right there. If you look at the codons, you count the codons, right? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. The anticodons that will associate in the transfer RNA with each codon, those are referred to as anticodons, are the 3 nitrogen-based sequence that is attached to the transfer RNA molecule. There are 20 amino acids that are attached to all of our transfer RNA molecules. There are 64 codons that are possible, because if you take the 4 different letters and you see all the possible 3-letter combinations, that's 4 to the 3rd power, that's 64. So they want you to use this codon chart to translate this messenger RNA. So we start with first GGC, and we go, starting here, G, so we're in this row, G, this column, so that puts us in the first letter G, second letter G, so we're right here, okay? And then the last letter is C, so we got G, G, and then look, is it the first, second, third, or fourth one in this box? It's C, so it's the second one, so it's glycine. We put glycine. Second codon is this one right here, UAU, and so you go UAU, and that's tyrosine. And you keep on going and going until you reach the 5th one right here. So that's the 3rd, that's the 4th, and then look, UAG. If you do UAG, that's a stop codon right there. And so stop, we're done. So we have a very, very short amino acid sequence of only 4 amino acids. If you were to do, let's say the first one, GGC, using the circular codon chart, you start in the middle, G, and go outside. Where's the next one? G, and then C, and then that's glycine. So once again, you're still going to get glycine. Second one is UAU, that's alanine. UAU, oops. UAU, UAU, tyrosine. UAU, tyrosine, sorry, there you go. UAU, tyrosine, correct. So you'll get the same answers. So we're going to take for number 10, this nucleotide sequence here, and I already chopped it up into triplets, so all the triplets are right here on the left, so CGG's right here, and then CTA and so on and so forth. And across, you're going to see what will the messenger RNA codon that goes across from this be if it were to be transcribed, and then what is the anticodon that matches it. So if you have CTA, across from C will be G, and T will be A, and A will be U because it's the messenger RNA. And then the transfer RNA anticodon that associates with that will be GCC. Oops, not GCC. Sorry. It will be, that is going to be CUA. I don't know why that's that way. That is going to be CUA. So that is an error. So let me just correct that. There we go. Oops. There we go. That is incorrect. There you go. And the rest of these should be right. All that should be right. That is my makeshift screencast because I don't have my original computer. Apologize for all the errors.